Very good. Good morning or good afternoon, everyone. Welcome to the Spring 2026 Include Data Hub webinar. My name is Joaquin Espinosa. I serve as the leader of the Administrative and Outreach Core for the Include Data Coordinating Center. And today we'll start with a presentation from Dr. Huqing Li from the National Heart, Lung, and Blood Institute, which is the institute funding the Include Data Coordinating Center, the Include Data Hub, and associated resources. And Dr. Lee will be doing a presentation, an overview of the Include ecosystem with some new and exciting resources being developed. Huqing, all yours.
Thank you, Joaquin. Good morning, everyone. Nice to see you today. Let me share my screen. Yeah, since I missed the last DCC fall webinar because of government shutdown, so today I will share with you some update from last fiscal year and some from fiscal year 26. First, to refresh your memory, I guess you're all familiar with the INCLUDE project. The mission for the INCLUDE project is to support research that investigate conditions affect individuals with Down syndrome and the general population. And it also aims to increase the number of investigators and the trainees studying Down syndrome. It also mission to work in partnership with the Down syndrome community to identify areas of needs and support work that benefit everyone. So in my presentation, I will cover the major update to you, the major INCLUDE events in the last few months. And I will update to you the INCLUDE funding for the last few years, and also update on data dashboard, on INCLUDE data ecosystem, and data NIH policies on data security and data privacy, protecting data privacy. Finally, I will update to you on the funding opportunities through the INCLUDE project. So this is a timeline of the key events under the INCLUDE project. In the last few months, the major milestones are here. So we had the DCC U2C award renew. We added a few new components to the existing DCC award, which includes the imaging data solution and the experimental model portal. And the experimental model portal was launched in October 2025. Additional components include the data security support and data analysis outreach, etc., like DOI, good implementation, and others. So the other major milestone for the INCLUDE project is the soft launch of the INCLUDE DSCDP common protocol, which just happened a few weeks ago. This figure is showing the NIH investment in Down syndrome research over the last 15 years. You can see the INCLUDE project dramatically changed the trend, the funding status for Down syndrome research at NIH. The green bars are the NIH non-include awards, and the orange dots are the total NIH Down syndrome funding. You can see there's a significant increase of Down syndrome research support at NIH. So by the end of fiscal year 25, INCLUDE project has funded more than 420 projects, which include 15 clinical trials and generated over 450 publications. This effort engaged 21 NIH institute centers and offices participation. The INCLUDE team had over 20 community engagement and outreach events. At the INCLUDE Data Hub, we have almost 16K individuals represented. As you may know, the INCLUDE project has 3 components. Component 1 is on basic science, component 2 is on cohort development, and component 3 is on clinical trials. From this figure, you can see that INCLUDE is expanding the investment in cohorts and clinical trial research. The INCLUDE Data Hub, the INCLUDE Data Hub is a product of the INCLUDE Data Coordinating Center. It's under INCLUDE Component 2. I want to share a few facts regarding to the Data Hub. First, it is a data repository. It's considered a controlled access data repository, which is called CADA. And the data storage is within NHLBI security boundary, and there's no PII. And the INCLUDE Data Hub is one of the 25 NIH best practice CADAs. We highly encourage you to deposit your data through to the Data Hub if you have INCLUDE funded awards. The genomics data is registered through dbGaP, and the data access request is through NHLBI Data Access Committee. So the INCLUDE Data Hub is also a human and experimental model data portal. So we have 2, now it looks like 2 separate portals. So for the human portal, data portal, we have large-scale data sets, clinical metadata. Those are harmonized clinical data. And for the experimental model portal, we have animal model information and also other experimental model information, including iPS cell lines. Through the INCLUDE Data Hub, we have tiered access, which covers open access for study, INCLUDE studies, cohort studies, and registered tiers through the INCLUDE data portal on individual level clinical data. And the controlled access applies to genomics data. The INCLUDE Data Hub is also a cloud computing platform, which is called Cavatica. Cavatica provides workflows and tools and interactive web apps for interactive data analysis. And INCLUDE product provide free cloud credit up to 20,000 to support your cloud computing. If you are interested, we have the information on the INCLUDE website, INCLUDE DCC website. So we just had the INCLUDE Data Hub just had the latest data release happened in May 2026, which include new study page of 13 studies included. And we also added additional participants and biospecimen data file information to the Data Hub. If you want to learn more, you can go to this website, which is on the INCLUDE DCC website under Help Center. So, over the past few years, we see researchers increasingly accessing INCLUDE data. And currently we had over 20,000 website visitors, and we have over 3,500 logins to the portal, over 3,000 Data Hub visitors and registered users. And currently we have 1,131 research profiles available through the INCLUDE Data Hub. And on the data access side, we have seen increased data access requests through dbGaP over the years. And we also see more and more secondary data users publish their research. The other major projects under Component 2 include the DS Connect registry, I guess you're all familiar with this, which facilitates information sharing among individuals with Down syndrome, families, clinicians, and researchers by collecting their demographic and health data information through surveys. The other major project is the INCLUDE Collaboration for Down Syndrome Progress, which was launched in fiscal year 2025, early 2025. It aims to enroll a cohort of individuals with Down syndrome across the lifespan and collect longitudinal data. As I mentioned earlier, we just had the soft launch of the CDP enrollment. So this figure is showing the structure of the INCLUDE data ecosystem, which include those major projects or programs under INCLUDE. So the data flows from either the experimental model studies or over 100 cohort studies. And in addition to the DS Connect Down syndrome registry, clinical trials, and the latest effort, great effort is the DS CDP, which include the DS clinical research sites and the DS clinical data coordinating center, DS4C, and the DS file repository data. So all those data will flow to the INCLUDE Data Hub, and after curation and harmonization, they will be made available through the Data Hub or the experimental model portal. And next, I want to update to you for a few major policy through NIH on protecting data security and data privacy. Those include guide notices. The first one is enhancing security measures for NIH controlled access data. And the second one is protecting human genomic data when developing generative AI tools and applications. And the third one is required security and operational standards for NIH cadres. Those guide notices will impact not only NIH cadres, including the Data Hub, but also the data access requesters. In order to meet the criteria of the CADA as controlled access data repositories, those efforts should be supported by NIH grant or cooperative agreement, other transaction and other funding mechanisms. And the INCLUDE Data Hub is through a cooperative agreement. And it also need to provide long-term storage for or provide access to data for research purposes. And it also has control access to data by prospective review of data access request through NIH DAC, and use federal employees to conduct reviews and authorized access. And the INCLUDE Data Hub meet all those criteria. And recently, the NIH implemented a strong identity and authentication assurance starting from April 1st, 2026. NIH cadres will be expected to adopt either the AAL2 and the IAL2, which means when you log into your controlled access data, you, in addition to IAL1, which are the ERA Commons credentials, there's another layer of access request, which is through either login.gov for domestic users or ID.me for international users. Currently, the INCLUDE Data Hub is under the IAL1 with acceptable compensating controls, and we are undergoing the process to move to the IAL2. So to put it all together, summarize all the three guide notices, effective April 2025, data access requesters from countries of concern are prohibited from accessing controlled access data. And effective March 2025, data access requesters training or developing generative AI models using human genomics controlled access data to treat the model and its parameters as data derivatives subject to the terms of access. What it means is if you are using controlled access to train your model and develop AI model, and those models will be considered controlled access as well. And effective April 2026, data access requesters requesting controls access data expected to meet NIST SP 800-171 standards or ISO IEC 27001 or 2702 standards, and be able to meet stronger identity and authenticator assurance levels. Yeah, if you want to learn more, please go to those guide notices. Okay, finally, we got some great news on funding opportunities. Include just published 3 RFAs. We got approval for those 3 RFAs. And the first receipt date is October 15th, 2026. The first one is the transformative R1. The second one is exploratory developmental research R21. And the third one is clinical trials phased awards R61, R33. All those 3 NOFOs covers 3 fiscal years. The first one, October 15, 2026. Second one is June 15, 2027. Third one is June 15, 2028. There's another RFA still available, still active, which is the training and career development F99K00. There's one last receipt date for this NOFO. If you are interested for those funding opportunities, I highly encourage you to reach out to your program officers to learn more. We don't have RFAs to support secondary data analysis, and which is very important for this group, I think. However, you can use the parent R21 or the parent R01 to support your data analysis. Well, we are waiting for the RFA to be released. I think that's all I have. Thank you very much. And let me know if you have any questions.
Thank you, Dr. Lee, for the presentation. A lot of exciting activities in the INCLUDE project. We do have time for questions. Any questions for Dr. Lee? And those, very excitingly, I'll be with you in a minute, Anita. You just heard our new RFAs for INCLUDE specific funding, R01s, R21s, clinical trials, with October 1st deadline, and then again next year in June, and I believe, which they are active until 2028 or so. So there'll be
3
multiple
years. Yeah.
3 years. Anita?
Thank you. I was going to put it in chat. Sorry. Thank you for the information. That was really helpful. Did you say, I just want to clarify something. Did you say that there is a possible NOFO coming out specifically for secondary data analysis that is under INCLUDE? Because I agree that's really important, considering all the new data that's coming out.
Unfortunately, it won't be under INCLUDE. However, I believe there will be a trans-NIH effort on that. Stay tuned. However, INCLUDE can support your, applications under the R21 or R1s through Select Pay. We will consider that if it's well received by the study sections. Very good.
And the other thing that maybe is worth clarifying, Huqing, is that if a trainee wants to apply to an F or a K award and they're doing Down syndrome research, they can apply through the parent FK mechanisms and then reach out to program officers, let them know that the application is there, and the Include project may be able to select that proposal for funding. Is that correct?
Yeah, if it's under the Include mission is on Down syndrome research, the Include project will consider any applications that is well received by a study section through Select Pay.
Great.
That's correct.
I think the difference, maybe for those who are not very familiar, the Include RFAs set up include specific study sections, right? Where every application in that study section would be Down syndrome research. Now you can still send an F or a K proposal, that may be assigned to a study section on immunity, neurology, or whatever the topic may be. But as long as you let the program officers know, that this is related to Down syndrome, Include may be able to go and do the Select Pay mechanism as Huiqing described. So
Yes,
please do turn in those. Yeah, do turn in those submissions, even if it is not an Include specific RFA.
That is correct. So if you have any additional questions, feel free to reach out to us. We can share more details on that. But the bottom lines, if your study is under Include mission, we will consider that.
And then, all of this information is in the NIH INCLUDE Project website.
We will update the website. That's a great point, Joaquin. We will update the website soon with all those new information. And specifically for the data sharing FAQ, we will update on that as well. And we, right. Of course, we highly encourage you to list Include Data Hub as your data repository. And we want the Include DCC to be the first entry for your data sharing because, we have other activities. We have the data harmonization process, and we have study pages for each study. We want your study to be represented on the Data Hub. Yeah, and we also have the GUID implementation, the DOI effort. So we want the researchers, Include research, to be fully aware of those. So that's why the DCC want to engage you once you get awarded. So
very good. Any other questions for Dr. Lee?
Yeah, feel free to reach out to me if you have any questions. Thank you.
Very good. If nothing else, then we'll transition to the next presentation, and this will be a short demo of some of the highlights of the Include DCC website and the Include Data Hub. But I thought it would be good to remind our friends in the community about some of the resources and then some of the key use cases of how to employ the Data Hub. And I'll go slow. If you want to follow me in your own browser, you're welcome to do so. What you're looking at right here is the website, the Include DCC website, which is separate from the Include Data Hub. And you can access through includedcc.org. So you put there in your browser, includedcc.org, you get here. A couple of things that I want to highlight for you, resources for the community. One is the Help Center with extensive documentation about pretty much everything in the Include Data Hub. So you can access that. I'm going to take a shortcut here. And something that I want to point you to is this resource library. You can also access that here. And the documentation. I have it open here. And the reason I think this is important is because if you ever get lost in the Include Data Hub, there are a lot of functionalities, a lot of potential slices and dices of the data of the cohorts. You can always come back here to the Include DCC and access the documentation that would explain step by step how to operate within the Include Data Hub. This could be from a quick start guide on how to log in, how to create an account, how to register for the Data Hub, how to connect across platforms. Dr. Lee explained clearly there are two important connectivities. One is with the Cavatica platform, the secure environment that provides access to control access data, but also to analysis tools, software packages that can help you analyze data. And the other one is dbGaP, the database for phenotypes and for genotypes and phenotypes. If you want to access control access data in the hub, you will need to request access through dbGaP for that study. Once approved, you'll be able to trigger that connectivity. So that's all explained here in the connecting platforms tool. And then we just talked about these various tiers of access. When you go to the data hub and you log in and you are asked to agree to terms of use, you are accessing what we call the register access tier, right? That is not enough for you to get access to the control access data. For that, you need, again, approval from dbGaP. And then dbGaP doesn't give you the data. The data is distributed through Cavatica. Important to keep all these parties straight. Dr. Lee talked about the release notes. If you come often to the data hub, you will see more things, more studies, more participants. Some studies may get more data. Some studies may add participants. So every time there is a release, you will find here a release note that explains what exactly is new relative to the previous release. So for example, you can see that recently in the past few weeks, we added new study pages. There were changes to some existing studies. There are new features in the portal, and you can go to the GitHub entry for the Include portal and see, the code for these features. And there may be updates to the data model that annotates every data element and the relationship across all these data elements. You can go see those in GitHub as well. Another tool that may be important, I don't want to assume that people know all of our acronyms, are these data dictionaries. So again, if you ever get lost and you want to understand what is different between a participant ID and an external participant ID, you can come here. If you want to know how do we call family units, what do we mean by a provan, a duo, a trio, you can come here and understand that, and so on and so forth. Same, with biospecimens, any aspect of the hub. What is a collection ID versus a sample ID versus an external sample ID, right? There's a lot of technicalities here. You may get lost when you're in the hub, not know the difference among these. Come back to the help center, to these resources. And then, of course, there is step-by-step guides on how to operate within the hub, from applying filters to how to use the participant tab. I'm going to give you a short demo of that now, how to use the biospecimens tab, how to use the data file tab. Great. So this is the help center. I want to make sure that you are aware of the help center. You can always come to office hours, but if you find yourself asking questions at any time of the day, you can come here to the help center. The other resource that I want you to be aware of is what we call the investigator registry. And this is here under Explore, Researchers and Publications. Researchers, every Include funded investigator eventually makes it to this investigator registry. So this is a great tool for you to find collaborators, to know who is being funded by the Include project to do X, Y, and Z. So you can find, for example, a particular individual, say Dr. Anna Esbensen and Cincinnati Children's Hospital. You click on that person, you can see the grants that the Include project has given to this person. You can see the publications that this person has produced with Include funding. And this is updated about once a year. So you should come and check your own profile. If you're an Include funding investigator, it's a semi-automated portfolio analysis. If we miss a publication and you want to make us aware of a publication, reach out to us, to our email address here in the Include DCC, and there are ways of manually entering or updating information. The other thing I want to highlight, this is also very useful for you to find people in your locality. Maybe you are a new trainee or a junior faculty, and you wonder, how do I apply to include funding? So you can find out whether in your institution there are any other include funding investigators. For example, here, Harvard Medical School, we have 5 investigators. You can look up for investigators. Maybe in Kansas, you can see different institutions in Kansas. So a great place to find collaborators, to find mentors, time and time again, redirected to those grants and to those publications. Great. Let's now switch gears and talk a little bit about the Data Hub. So that was the website. This is now portal includedcc.org. Now, before you log in, there's quite a bit of information that you can peruse and review without having to set up an account. Something that has happened relatively recent, is a recent development in our journey here, is that we have made the study pages public, meaning you don't need to log in to see the study pages. So I want to explain a little bit what this is. Every include funded investigator that has a human research component, in other words, an IRB protocol, is expected to contribute that data to the Include Data Hub. So this is the component 2 that Dr. Lee was talking about, all these cohort studies. To give you a sense of the scale, there are more than 50 include grants that have various human research components, and that number probably grew. The number 50 is from last year. In the last 12 months, probably we got many more. All of those eventually need to be represented here. So I want to clarify about what's found in this table. You know, the studies are listed alphabetically. We're going to be clicking on some of these. Most of them are from the Include program. There are a few from the Kids First program. And then you will see this, we'll talk about this column DB gap. This will indicate whether the study has control access data. But I want to dwell on this H and the U. If you see a study that has an H, means that the data, the clinical metadata, demographics and clinical metadata from that study has been curated, harmonized, mapped to ontologies. You will be able to find those participants in the data exploration tab, okay? If you see a U for unharmonized, it means that the data is available as the data generator provided it, but we haven't gotten yet to the step of harmonizing it, mapping it to medical ontologies. You will not find that study in the data exploration tab, and we'll make that more obvious in a moment. And then there's one other letter that I want to highlight, shown here, which is this G. The G stands for NDA GUIDs. These are global unique identifiers. This is something that we want to encourage everybody to use. These are done through the National Data Archive of the National Institute of Mental Health, where you can mint a unique identifier for your participants. That would allow you then to see if your participant is present in other studies. Without a GUID, you wouldn't be able to know whether some DS Connect participants are also in the DS RRS study. GUIDs are critical to be able to identify that overlap across participants from many studies. Great. So I'm going to log in. I could click here on the studies and start showing you the studies from the public tier, but I want to log in and show you the studies from the registered tier access. Here we are. I want to highlight the various possible entry points to this massive data set. We'll go to the studies. You can see studies. You can skip the studies and go to participants. You can go to biospecimens for the studies that have biospecimens, or you can go straight to data files. An analogous way of thinking about it is in data exploration. You can explore the data by looking for participants of certain characteristics. Or you may be interested in securing biospecimens. You can start looking for biospecimens and then filter by studies of participants. Or you may be already interested in some specific data files, and then you'll find the data files and then filter by participant characteristics. So going back to studies, I want to show you a couple of these studies and highlight a couple of features. Again, every include-funded study will eventually have one of these. Some studies will have participants and files, may not have biospecimens. Some studies will have biospecimens. The top box here is the study page, where you get to understand what this study is about. If it is a clinical trial, you may have an entry in clinicaltrials.gov. You will see a description. You will see the publications associated with this study. And then you can, if you want to learn more about this study, you can navigate away to PubMed and look at the publications being associated with this study. Okay? And a study may be a single grant, and a study may be more than a single grant. Maybe that a group is studying a cohort of people with Down syndrome, and they are funding that through multiple grants. So sometimes the studies have a one-to-one relationship to grants, sometimes they don't. Now, because this study is harmonized, you'll be able to see that then there are summary statistics using the two ontologies, key ontologies that we use here in the Data Hub, the human phenotype ontology and the Mondo disease ontology, okay? And you can see the various rates of co-occurring conditions for the participants in this one study. Because there is harmonized data, you'll be able to also see the demographics. This happened to be all participants with trisomy 21. If you ever come into a study and you see these boxes empty, it's because they haven't been harmonized yet. It doesn't mean they don't have data. It just hasn't been harmonized yet. I want to show you another study to illustrate more other features when you do have biospecimens or when you do have control access data. When you see in a study this DVGAP accession number, it means that this study has control access data. By simply logging in the Data Hub, you will not be able to access that data. You will need to request approval through DVGAP. So if I click on that study, it will take me now to DVGAP, right? Genotypes and phenotypes. This is the entry for the Human Trisom Project. And then I can now request access to these data sets. And Dr. Lee will get that request, review your proposal, approve it, hopefully. And then I will show you what that does once you come back to the Data Hub. Great. So for studies that already have data sets, after the study section in the study page, you will have the data set section. And it's a particular study that has various data sets. You can see whole genome sequence, TCR-seq, whole blood RNA-seq. Now, some data sets can be harmonized, meaning we can use a common pipeline to align the reads to the genome, or we can use a common pipeline to do the RNA-seq process data. That's why you see this harmonized. Some data sets are one-off, maybe the only type, the only data set of that type in the Hub, there hasn't been a need to harmonize it yet. That's why you will see this unharmonized, right? It means that they haven't been matched in terms of processing to other studies. Great. In the last few minutes, I want to come back here then to the concept of data exploration. I want to clear this filter that I was setting up earlier. And briefly introduce the three main entry points to the massive multidimensional data set of the Data Hub. You can start with participants. You may be looking for individuals with certain characteristics. Again, not all studies have made it to this section yet. If they are not harmonized, they will not be showing up here. Only those that are harmonized are showing up here. Say that you don't care which study is coming from, you want to select all of them. Of course, you get to select whether you want controls or only trisomy 21. You can apply that. Can apply these to make sure I get all the studies. And then there are two different ways of exploring the clinical metadata. And you may have a favorite ontology or not. You can try it both ways, see what happens when you look for participants with a certain condition with one versus the other one. Say that you're looking for participants with hypothyroidism, then you will apply that filter. I want to show you what has happened here. Now I have all the studies selected. I'm interested in those with trisomy 21, but also euploid controls, only those with a mondo diagnosis of hypothyroidism. Now my universe has shrunk significantly. So now I have my participants, but I also see the biospecimens associated with those participants and the data files associated with those participants. I can keep going and keep filtering, right? I want to make sure that you are aware of the various ways in which we treat age. Age at diagnosis means that we have a value for when that person was diagnosed with hypothyroidism. That may not be the age of the participant at the time of the biospecimen collection that then led to that data file. Let me say that again. The person may have been diagnosed with hypothyroidism at 5 years old, but the age at the observed phenotype may have been later on. The study met the participant when the participant was 10 years old, and that may have been when the blood sample was collected. And of course, you can filter by demographics, is that we apply a filter here for females, and then we can apply a filter here, say, for Black or African American. And then as you see, as you apply more filters, your universe keeps shrinking, and so do your biospecimens and your data files. Now, I want to talk about the data files for a moment. Let me check my timer. I'll be wrapping up here. R is register, but I'm not authorized. And register meaning I will have access within the register tier access just by having an account in the DCC. But unauthorized means that I will need to request access to access that data through the big app. This exclamation point, you can zoom in there and see. I don't have an authorization status. I cannot download this data. So if I want to access this one file for this participant, I will have to request access to the big app. What will happen once I have that, if we go back for a second, that lock will become open. And then that means that I will be able to get access to that file. How am I doing here? I want to just use one other minute because Dr. Lee talked about the experimental models portal. And this will take you slightly away from the data hub. It looks very similar. It feels very similar. But now we're no longer dealing with human research studies. Now we're dealing with studies that have used animal models or cell-based models of Down syndrome. We had a full demo of these experimental models of Down syndrome portal in the fall webinar. Just want you to be aware of it. If you have any questions, you can always ask. But this is the latest research developed by the INCLUDE DCC to support what we call the component 1 investigators, those investigators doing basic studies of trisomy 21. I will stop here with the demo. Hopefully that was some of the highlights of the INCLUDE DCC website, the data hub, and then of course you can always show up to office hours, access the help center, and or just send us a ticket and we'll engage with you directly. Let's see if there are any questions about this. I want to keep my presentation brief. We have two very exciting scientific presentations today. Any questions? So it's my pleasure to introduce Micah Donovan, an instructor of pharmacology at the Cynic Institute for Down Syndrome at the University of Colorado, who will be presenting on a new data analysis on data sets from the Human Trisom Project. Micah, do you want to share your screen? Let's confirm that.
Yeah.
We can see your slides.
All right, so today I'll be presenting some of the results from a recently published paper that we called the Systemic Multiomic Deconvolution of the Clinical Heterogeneity of Down Syndrome, where using transcriptomics, proteomics, metabolomics, and immune profiling from one of the largest deeply phenotyped Down syndrome cohorts assembled to date, we asked whether common co-occurring conditions in Down syndrome are associated with distinct molecular biosignatures. Now, before I get started, I'd like to acknowledge the team that made this work all possible. Specifically, I want to recognize Sarija, who served as the lead analyst on this project and was central to the integration and interpretation of these large multiomic data sets. I'd also like to thank Belinda and Kaylee, who were instrumental for much of the clinical data curation, making these analyses possible. I'd like to also acknowledge our funding agencies, namely various NIH institutes, which provided funding support for data generation, in addition to the Global Down Syndrome Foundation. Finally, and most importantly, I'd like to thank the individuals with Down syndrome and their families who participated in this research. Before we dive in, here's a brief roadmap for today's presentation. I'll start with an overview of the clinical diversity represented in the cohort, and I will explain the study design that fueled our discoveries. Then I'll walk through several examples of how common co-occurring conditions of Down syndrome associate with distinct molecular signatures. Finally, I'll show how these different clinical phenotypes uniquely reshape the canonical biosignatures that we typically associate with Down syndrome, before closing with some key conclusions and future directions. Let me get my laser pointer out here. Okay. Now, it's important to appreciate the clinical heterogeneity of Down syndrome. Although trisomy 21 is the common underlying genetic cause, the clinical presentation is remarkably diverse. Individuals can experience a wide range of co-occurring conditions, and no two clinical profiles are exactly alike. While conditions such as congenital heart defects, hypothyroidism, and sleep apnea are among the most common phenotypes in our cohort, many other conditions occur across a substantial subset of individuals. And interestingly, this diversity extends even within specific disease categories. For example, among participants with congenital heart diseases, we observe multiple distinct cardiac defects rather than a single dominant diagnosis. Now, this raises the question, how is this heterogeneity distributed across the population? In other words, are most individuals affected by just a few conditions, or do some accumulate many different clinical diagnoses? Here we're looking at how these phenotypes are distributed across individuals within the population. Now, the first thing that becomes apparent is the remarkable variability in clinical complexity. In the heat map on the left, each row represents an individual, and each column represents a clinical phenotype. While some individuals have relatively few documented conditions, others accumulate many different co-occurring conditions. In other words, there's no typical clinical profile of Down syndrome. In fact, we see a broad distribution in the number of co-occurring phenotypes per individual. Some participants have very few conditions, while others have extensive clinical complexity. Now, what's important to understand is that this variability does not appear to be driven simply by age. In fact, we observe very little relationship between age and the total number of clinical phenotypes, suggesting that medical complexity is influenced by factors beyond simply getting older. Finally, if we jump back to the heat map, we can begin to see how certain conditions tend to occur together. Rather than being randomly distributed, some phenotypes cluster within subsets of individuals. To explore that further, we examined the clinical associations between the co-occurring conditions. What we're looking at here is how examples of individual clinical phenotypes associate with other conditions within the Down syndrome population. What becomes clear is that co-occurring conditions are not randomly distributed, and that each phenotype exhibits a distinct pattern of clinical associations. Now, some of these relationships are intuitive and recapitulate relationships that are well established in the broader population. For example, obesity being strongly associated with sleep apnea and high cholesterol. On the other hand, some relationships are less obvious. For example, obesity is associated with conditions such as hearing loss, and hypotonia shows associations with several cardiac and developmental phenotypes, including patent foramen ovale and patent ductus arteriosus, which are two forms of congenital heart defect. Importantly, the associated phenotypes differ depending on the condition being examined. In the case of obesity and hypotonia, these conditions do not share the same clinical network of associations, suggesting that different co-occurring conditions may define distinct clinical subgroups within the Down syndrome population. Ultimately, this clinical profiling raises an important question that motivated the rest of our work. It's, do these distinct clinical phenotypes correspond to distinct molecular and immunologic signatures? To address this question, we performed a systematic multi-omic deconvolution of the clinical phenotypes in Down syndrome. Our cohort consisted of 356 individuals with Down syndrome and 146 euploid controls spanning a broad age range. Now, for each participant, we collected extensive clinical information encompassing approximately 100 clinical phenotypes. We then integrated 5 complementary biological data sets, including whole blood transcriptomics, plasma proteomics, plasma metabolomics, immune cell profiling, and circulating immune markers. In total, this provided roughly 35,000 clinical data points and approximately 22,000 molecular and cellular features. We systematically tested within the T21 cohort associations between every clinical phenotype and every molecular feature, comparing cases versus controls, which resulted in more than 2.2 million association tests. And this framework allowed us to examine what molecular and immunologic signatures are associated with individual clinical phenotypes. It also allowed us to query which biological layers show the strongest associations for specific conditions. Now, one phenotype that stood out immediately was obesity. When we ranked the clinical phenotypes by the number of significant molecular associations, obesity was associated with the largest number of proteomic changes and the largest number of metabolomic changes of any phenotype we examined. Now, it's important to understand that this doesn't necessarily mean that obesity is the most important clinical condition in Down syndrome. Rather, it suggests that obesity is associated with particularly widespread systemic biological alterations that are readily detectable across multiple molecular layers. And these volcano plots show the widespread remodeling of both the plasma proteome and metabolome in obese individuals compared with non-obese controls in the Down syndrome cohort. Perhaps unsurprisingly, many of the strongest proteomic associations are biologically intuitive. For example, we observed alterations in adiponectin, an adipose-derived hormone that regulates insulin sensitivity, leptin, an adipose-derived hormone that regulates appetite, and we also observed changes in inflammatory and immune modulators such as C-reactive protein, serine amyloid A1, IL-6, and other molecules involved in energy balance, metabolism, and inflammatory regulation. Now, among these signals, leptin and insulin, shown here on the right, stand out. Obese individuals exhibit significantly elevated levels of both molecules compared with non-obese individuals with Down syndrome and also euploid controls, indicating that obesity and Down syndrome is accompanied by substantial endocrine and metabolic dysregulation. However, the molecular signature of obesity extended beyond classical metabolic regulation. In addition to the altered metabolic signaling, obesity was associated with evidence of systemic inflammation exacerbated above the baseline level observed in the Down syndrome population in general. For example, both IL-6 and C-reactive protein were significantly elevated in obese individuals, consistent with chronic low-grade inflammatory activation. We also observed changes in proteins linked to liver physiology. For example, aldehyde dehydrogenase 2, which is a mitochondrial detoxification enzyme and a marker of oxidative and metabolic stress. ALDH2 was increased in obese individuals relative to non-obese controls, whereas albumin levels were reduced. Ultimately, these findings suggest that obesity in Down syndrome is not limited to adipose tissue and glucose regulation, but also includes inflammatory pathways and markers associated with hepatic stress. The metabolomic data also provided some additional insight into the biological pathways altered in obesity. First, we observed evidence of disruption of the glutamate-glutamine cycle. Obese individuals exhibited higher glutamate levels and lower glutamine levels, suggesting altered amino acid metabolism in this cycle. Second, we identified changes consistent with dysregulated mitochondrial lipid metabolism. Both octanoyl carnitine, a fatty acid transport metabolite, and 5S-HETI, an inflammatory lipid metabolite derived from arachidonic acid metabolism, were elevated in obese individuals, implicating altered fatty acid utilization and lipid signaling pathways. Now, this raised an important question. How much of this biology reflects obesity in general, and how much may be specific to the Down syndrome population? To address this, we compared obesity-associated molecular changes in individuals with Down syndrome to obesity-associated changes observed in the euploid controls. If we look at these scatter plots here, we have the changes in the Down syndrome population on the y-axis and the euploid control population on the x-axis. Overall, we observed concordance between the two populations. Many proteins and metabolites changed in the same direction in both groups, indicating that much of the molecular biology of obesity is shared between individuals with and without Down syndrome. However, the relationship was far from identical. The magnitude of change varied considerably across features, and a number of proteins and metabolites were significant only within the Down syndrome cohort, as shown by the color coding notated below here, with blue being significant in T21 only, pink being significant in D21 only, and orange denoting those features that were significant across both karyotypes. Now, examples include IGF-BP1 in the proteome and glutamine in the metabolome, which exhibited distinct effect sizes compared with the euploid population. Taken together, these findings suggest that obesity and Down syndrome is characterized by a combination of shared and unique biology. Many core obesity pathways are preserved, but trisomy 21 appears to modify specific responses to increased adiposity. When we examined the transcriptomic associations across clinical phenotypes, congenital heart defects emerged as one of the strongest signals in the entire data set. In particular, atrioventricular canal defects, or I may refer to them as AVC throughout the next couple of slides, were associated with the largest number of differentially expressed genes in the whole blood transcriptome, exceeding all other congenital heart defect subtypes and most other clinical phenotypes. In the heat map on the right here, this illustrates these transcriptomic changes across the individual congenital heart defect categories. Now, what stands out in this heat map is the striking gene expression signature associated with AVC in the top row here, whereas other cardiac phenotypes exhibit substantially weaker effects. Now, an important takeaway here is that this signal is not explained by prior surgical correction, denoted by CHD repair in this row here. Individuals with a history of CHD repair do not exhibit the same transcriptomic pattern of that of AVC, indicating that the observed changes are associated with the underlying phenotype rather than simply a consequence of surgery. Now, when we examine the magnitude and composition of the transcriptomic changes, what immediately stands out is the scale of the effect, with approximately 1,700 differentially expressed genes for ABC in the whole blood. And you can appreciate the breadth of these changes by looking at the volcano plot in the middle here. We observe hundreds of genes, both upregulated and downregulated in individuals with a history of ABC, including genes involved in innate immunity, inflammation, extracellular matrix remodeling, and neutrophil biology, which ultimately encompasses roughly 10% of the measured whole blood transcriptome being significantly altered in individuals with a history of ABC, indicating that there is broad transcriptomic remodeling rather than a perturbation of a small number of genes or pathways in individuals with a history of ABC. Now, pathway analysis revealed a striking pattern here. Specifically, many of the pathways already known to be dysregulated in Down syndrome, shown in the left on this heat map here, were further amplified in individuals with a history of atrioventricular canal. These included inflammatory and immune pathways such as the interferon gamma signaling response, which is the single most elevated gene signature in individuals with Down syndrome. We also observed enrichment of pathways related to hypoxia and heme metabolism being the second most enriched gene signature in Down syndrome. And the heat maps in panel B here show representative genes underlying these signatures. Genes involved in inflammation and innate immunity, which again show increased expression in Down syndrome and further elevation in individuals with ABC, and a similar pattern observed for genes involved in heme metabolism and hypoxic responses. Now, these specific examples illustrate this phenomenon directly. For example, both IFI27, an interferon-stimulated gene associated with antiviral immune activation and inflammatory signaling, and ALAS2, an enzyme involved in heme biosynthesis. Both of these genes are elevated in Down syndrome relative to euploid controls, but are elevated even further in individuals with a history of ABC. Together, these findings suggest that ABC does not introduce an entirely new molecular program. Rather, it appears to exacerbate existing inflammatory, immune, and hypoxia-associated signatures already present in Down syndrome. We next asked whether these transcriptomic differences were accompanied by changes in biological levels beyond the transcriptome, and the answer was yes. Many of the immune cell abnormalities that are already observed in Down syndrome were further accentuated in individuals with a history of ABC. Specifically, individuals with ABC showed further reductions in both CD4 and CD8 naive T cells, accompanied by increases in more differentiated T cell populations, including central memory CD4 cells and CD8 TEMRA cells, suggesting that a history of ABC in Down syndrome is associated with more pronounced dysregulation of T cell homeostasis and maturation. We also observed elevated levels of multiple circulating proteins associated with cardiac stress and tissue remodeling in individuals with a history of ABC. Several of these markers, including natriuretic peptide B, collagen 1A1, and tropomyosin 1, were not significantly altered in Down syndrome, but were specifically elevated in individuals with atrioventricular canal. In contrast, markers including angiopoietin 2, thrombospondin 2, and NGF are elevated in Down syndrome, but even more so in those with a history of ABC. And again, this can be appreciated by looking at the individual data points plotted on the right here. Now, one remaining question was whether these signatures simply reflect early life consequences of congenital heart defects, or whether they persist throughout the life. To address this, we examined representative transcriptomic, proteomic, and immune cell features across age. The key observation is that in many cases, the differences associated with ABC persist throughout the lifespan. While many of these biomarkers change with age in Down syndrome as a whole, individuals with a history of ABC remain consistently shifted relative to individuals without ABC. And you can see this with an example, some select examples of genes here, the natriuretic peptide B here, and CD4 naive T cells here. These findings indicate that many of the molecular and immunologic consequences of ABC are not transient developmental effects, but instead represent persistent biosignatures that remain detectable from childhood through adulthood. Now, shifting our attention, neurological phenotypes also exhibited distinct molecular signatures. When we examined proteomic associations across neurological conditions, a history of generalized seizures emerged as the strongest signal, with nearly 500 significantly altered plasma proteins. This substantially exceeded the signatures observed for other neurological phenotypes, including autism spectrum disorder, hypotonia, regression, cognitive decline, fatigue, and nystagmus. Notably, approximately 10% of the measured plasma proteome was significantly altered, indicating widespread biological remodeling associated with a seizure history. And the heat map on the bottom here shows how these different neurological conditions do not share identical molecular profiles, perhaps unsurprisingly. Each phenotype exhibits its own pattern of proteomic alterations, suggesting that distinct neurological manifestations in Down syndrome correspond to distinct underlying biology. Examining the proteins driving the seizure-associated signature, we found it to be enriched for proteins directly related to neural structure and synaptic function. Several of the strongest associations involve proteins linked to neural cell adhesion and neurite outgrowth, suggesting alterations in neuronal connectivity and network organization. We also observed changes in extracellular matrix proteins, indicating potential remodeling of the neural microenvironment. And perhaps most notably, proteins involved in synaptic biology were elevated in individuals with a history of generalized seizures. Examples include oligofrenin 1 and clathrin interactor 1, which both played important roles in synaptic vesicle trafficking and neuronal signaling. Turning our attention to gastrointestinal conditions, what immediately stood out was that GI conditions were associated with robust molecular signatures across multiple biological layers. However, these signatures were not uniform across the conditions. Some phenotypes showed their strongest signals in the plasma proteome. Others were dominated by transcriptomic changes, while others were primarily reflected in metabolite or immune cell profiles. That heterogeneity is evident in the heat maps below. Although all of these conditions fall under the gastrointestinal umbrella, the sets of associated proteins and transcripts are largely distinct. In other words, gastrointestinal conditions in Down syndrome do not share a single molecular signature. Each condition appears to have its own characteristic biological fingerprint. One example is celiac disease. Unlike some of the other GI conditions, celiac disease showed coordinated changes in both the plasma proteome and immune cell compartment. On the left are some of the strongest protein associations. Many of these proteins are linked to immune activation, including CXCL1, LAG3, CD8A, or CD274, also known as PDL1, suggesting that ongoing T cell activation and immune stimulation is altered in individuals with celiac disease. As was the case for several of these molecules, represented by, or a representative example shown here is CXCL1 and CD274, these factors are elevated again in Down syndrome, in the Down syndrome population in general, but even more so in individuals with a history of celiac disease. We also observed clear shifts in B cell differentiation. Individuals with celiac disease had increased plasmablasts and increased IgM memory B cells, indicating altered humoral immune activity in addition to T cell activation. And again, both plasmablasts and IgM memory cells are elevated in Down syndrome in general, but more so in celiac disease. GERD, on the other hand, presented a different molecular profile. Unlike celiac disease, GERD was dominated by transcriptomic and metabolic alterations. In the transcriptome, we observed exacerbated overexpression of multiple genes involved in heme metabolism and erythroid biology, as demonstrated by RAP1 gap here, a factor involved in cell adhesion, barrier integrity, and cellular signaling dynamics. Again, these factors are elevated in Down syndrome in general, but exacerbated in GERD cases relative to controls. The metabolome told a complementary story. Several circulating amino acids, which are depleted in Down syndrome, were further reduced in individuals with GERD, as demonstrated by proline here. Together, these findings reinforce the idea that individual GI conditions in Down syndrome are associated with distinct biological pathways and molecular phenotypes. I'm going to speed through the next little bit here because it looks like I'm running out of time. Next, we wanted to define a more complete picture of how the broader biological pathways that define Down syndrome vary across clinical phenotypes. On the far left here, we see the canonical transcriptomic signature of trisomy 21 compared to euploid controls, including elevation of interferon signaling, inflammatory responses, heme metabolism, oxidative phosphorylation, and other hallmark features of Down syndrome. When we asked how these same pathways behave within the Down syndrome population when individuals are stratified by co-occurring conditions, what emerged was substantial heterogeneity. The magnitude of pathway enrichment clearly differs across clinical phenotypes, despite all individuals sharing the same underlying chromosomal alteration. Some conditions show further enrichment of pathways elevated in trisomy 21, while others show attenuation or even depletion of these same signatures, suggesting that co-occurring conditions are not simply clinical labels layered onto a common molecular background. Rather, they are associated with distinct modulation of the core biological programs that characterize Down syndrome. For example, when we rank co-occurring conditions by the enrichment of interferon gamma response signatures, the most enriched transcriptomic signature in Down syndrome, we see that the pathway is not uniformly elevated across phenotypes. While dysphagia shows the highest enrichment of the interferon gamma response, indicating further enrichment beyond the baseline trisomy 21 signature, overweight sits at the opposite end, showing relative attenuation or depletion of this same pathway. And the heat map in the middle here shows that this enrichment is not driven by a single transcript, instead multiple interferon-stimulated genes, which are elevated in Down syndrome, shown in the red color scale here, and are further elevated in individuals with dysphagia. And some representative examples illustrate this phenomenon here. Skip a slide here. Another example is the heme metabolism transcriptomic signature, which is second only to the interferon gamma response signature in terms of enrichment in Down syndrome. Again, when we rank the clinical phenotypes by pathway enrichment, we see substantial heterogeneity around that baseline trisomy 21 signature. Reactive airway disease falls at the top end of the distribution, again, indicating further enrichment of this signature. whereas PFO, a congenital heart defect, falls at the opposite extreme and shows relative depletion of the pathway. And again, these transcripts are elevated in Down syndrome overall, but further increased in individuals with reactive airway disease, but not so in PFO. Now, ultimately, these examples demonstrate that co-occurring conditions do not simply add new biology on top of Down syndrome. Instead, they can amplify or attenuate the very pathways that define the Down syndrome molecular landscape. So after all of this, I hope to leave you with some key conclusions. Clinical phenotypes in Down syndrome are accompanied by distinct multi-omic biosignatures spanning gene expression, circulating proteins and metabolites, and immune cell composition. Some co-occurring conditions are characterized by widespread molecular remodeling, whereas others produce more focused alterations restricted to specific biological domains. Canonical Down syndrome pathways are not altered uniformly across individuals, and that co-occurring conditions can amplify, attenuate, or uniquely reshape these established molecular signatures. And ultimately, these findings support a shift from viewing Down syndrome as a single molecular entity towards understanding biologically distinct subgroups that may benefit from personalized monitoring and therapeutic strategies. Finally, I'll finish with 3 questions that I think emerge naturally from this work. First, the chicken or the egg question. Are these molecular differences contributing to the development of clinical phenotypes we observe, or are they consequences of those phenotypes? The reality is likely some combination of both, but distinguishing cause from effect will require longitudinal and mechanistic studies. Second, the special or shared question. We showed that obesity, congenital heart defects, celiac disease, seizures, and other conditions have distinct molecular signatures within Down syndrome. But how much of that biology is unique to Down syndrome, and how much reflects the mechanisms that are shared with the broader population? Understanding that distinction will help us identify both Down syndrome specific pathways and opportunities to leverage existing knowledge from other fields. Finally, the bang for our buck question. Across these analyses, we identified hundreds to thousands of altered genes, proteins, metabolites, and immune features. Which of these are merely biomarkers and which are actionable targets? Which signatures provide the greatest clinical value for risk stratification, monitoring, or therapeutic intervention? And I believe that answering these questions will be essential if we want to translate these molecular observations into meaningful precision medicine for individuals with Down syndrome. And with that, I will take any questions.
Hey, thank you, Micah. Very good. Yeah, we do have time for questions, because we'll go to the top of the hour, so have plenty of time for Anita as well. Any questions or comments for Dr. Donovan?
Yeah, I'll raise my hand. So thank you so much, Micah, for the great presentation. It's very interesting research. So I have a few questions regarding CHD. The first question, I just want to clarify for the inflammatory immune and hypoxia signaling check in Down syndrome with CHD. Did you also check the congenital heart disease repaired population, or the data you showed already included that? So I just want to make it clear if the heart repair affect the those signal pathways or not. Second question related to that is for the CHD transcriptome study again. So which data point, how many data points did you check, and what kind of age range you check those? I have another one, but I want you to address those first.
Sure, yeah. So to answer your first question, I can actually go to the slides that'll help with the contextualization here. So regarding the question about whether we checked the CHD repair, that was included in our analysis. And surprisingly, there were not very many transcriptomic alterations when you look at cases versus controls, so people that had CHD repair versus those without CHD repair in the transcriptome, right? So up here, you can see all of the changes associated with ABC in this row, but you can see here that CHD repair had very few transcriptomic differences between those that did not have CHD repair. Now, taking it a step further, comparing whether or not people that had ABC that had CHD repair versus people that had ABC without CHD repair, we did not ask that question. I would speculate that we weren't necessarily powered to do that, because once we start breaking up the cohort into all these different clinical phenotypes, we may not have enough power to make those comparisons. But I think it's a valid question, and something that remains on our mind is, whether or not CHD repair in ABC is distinct from ABC without CHD repair. And your second question was about how many people did we analyze?
The data point for the transchromosome study in the following a few slides.
Yeah, so we have about 16,000 genes in our RNA sequencing. And so for each of the clinical phenotypes, we broke it down into cases versus controls and looked at the differential expression across all 16,000 genes. So I believe that our number of individuals with AVC in our cohort, correct me if I'm wrong, Joaquin, was
In the
about
next slide, Micah. I think the sample sizes are probably in your next slide.
Data point across the lifespan. So what stage?
Across the lifespan, yeah.
33. It was there, 33 cases. Yeah.
Okay.
Yeah,
okay,
so we had 33
1,
cases
2, 3.
of ABC. Yeah, and so these slides or these plots here, show the age on the x-axis, and the red is the ABC cases. The green is the T21 without ABC, right? So that's the 33 versus the remainder of the T21 cohort across age.
Okay, thank you. So another question is, you didn't check the whole genome sequencing about gene mutation, change between congenital heart disease, Down syndrome with or without congenital heart disease. Have you done that, or are you planning to do that?
We have not done that. I do believe we are planning on getting deeper into our analysis of whole genome sequencing. But to date, we haven't really looked at, differences in clinical associations with regard to, whole genome sequencing.
Thank you. That will be very interesting to identify those modifier genes.
Yeah, just a quick comment there, though. 33 cases isn't going to be very well powered for the genomics unless we've got really, well-known variants there, right? You'd have to select certain variants.
I'm sorry, Matt. What did you say?
Sorry, I don't know that we'd be very well powered for any, genetics associations with just 33 cases, right? This is why we need things like the cohort development program, right?
That's true. We need
larger cohorts. Yeah.
Thank you.
Thank you.
Well, thank you, Micah.
My pleasure.
For the presentation. Yeah, we'll transition here to our second scientific presentation of the day, and it's my pleasure to introduce Dr. Anita Bhattacharya from the University of Wisconsin-Madison, who will be presenting on some very interesting work that was recently published, I believe, in Science, Multiomics of Down Syndrome Early Brain Development. Anita, all yours.
Wonderful. Well, I'm really happy to be part of this, so thank you for the invitation. And I will say it's hard to follow Micah's talk. That was super interesting. And it's interesting, and I'll come to this at the end, that I think we have the same outstanding questions. So that's really great. My talk is going to be focused specifically on the brain, and my lab has been working to understand how brain development is altered in Down syndrome for many years. And I think we all appreciate that Down syndrome is characterized by, it impacts the entire lifespan. We think of it as, many people think of it as divided into two separate processes of altered neurodevelopment and then increased neurodegeneration. And we know that the developmental deficits lead to structural and functional differences that are obvious at birth in babies that are born with Down syndrome. And then we also know that neurodegeneration, Down syndrome, associated Alzheimer's disease but when we think about the data that's available for looking at the molecular neuropathology across the lifespan, the data that we have, whether it be cellular data, whether it be omics data, whether it be clinical data, is generally fragmented across developmental windows and across data sets. And so we have some information. Sorry, let me get my laser pointer. We have some information about prenatal development when the brain is forming, although that is limited by accessibility. We have a lot of clinical information about adolescents, adults, and older adults. Much of that is focused on Alzheimer's disease as well as other co-occurring conditions. And so we feel like, at least since thinking about the brain, we have a gap here. And so what our research team decided to do was to look at two really critical areas of the brain at birth. And I'll tell you a little bit more about them, and I'll just want to acknowledge my really fantastic collaborators, Daifang Wang, Andre Sosa, and Sushan Zhang. And so we looked at basal forebrain and the prefrontal cortex from in postmortem tissue from 0 to 3, so right after birth. And so the first thing I want to tell you about is the basal forebrain. We chose the basal forebrain because, for those who may not realize this, the basal forebrain is a very specific part of the brain that is prone to degeneration in aging and in several neurodegenerative diseases, including Alzheimer's disease and Down syndrome. And the basal forebrain cholinergic loss, the loss of the cholinergic neurons in this area, is a hallmark of both cognitive decline and memory loss. So the number or the density of basal forebrain cholinergic neurons actually correlates with memory, and especially in aging. We also know, due to some recent data, that the basal forebrain volume decreases in individuals with Down syndrome. As you can see by these plots, both the anterior medial and the posterior basal forebrain decrease at about the time coincident with DSAD in individuals with Down syndrome, and they do so much faster than the controls. And so what we did was we took, again, postmortem basal forebrain tissue from 4 control and 4 Down syndrome individuals, and we did single nuclei RNA and attack sequencing. And we were a little surprised after we got the data to learn that this was the first sequencing of any human basal forebrain. This is an area of the brain that people don't really look at a cellular and molecular level. There's a lot of imaging studies, but the imaging studies often miss this area. So we had, as I said, the age of these individuals was 0 to 2 years old, and we tried to match them for age, sex, and PMI, and we had 4 of each. And this was work that was largely carried out by Nicole West, who is a recent graduate of my lab, and Kalpana, our college, who is our bioinformaticist. So when we look at the data, and I'm going to present mostly the transcriptomics, we're still working to understand a lot of the attack data. When we look at the transcriptomics, we see all of the cell types that we would expect to see in the basal forebrain, which is the inhibitory neurons, the basal forebrain cholinergic neurons, and then non-neuronal cells. And what we were initially surprised to see is when we look at the composition in control and Down syndrome samples, we see more neurons and less astrocytes. And this is contrary to what most people think about when they think about the cell composition in the brain. And I'll come back to that again later. When we look specifically at the basal forebrain cholinergic neurons, these neurons that are prone to degenerate in Down syndrome and in Alzheimer's disease, we were surprised to find, although we had a small number of these cells, they clustered very clearly into two subtypes. And based on gene expression or marker gene expression in these 2 subtypes, we concluded that they are mature and immature cholinergic neurons. And remember, this is from tissue at birth, and so the cells are still developing. They have turned into neurons, but they're still maturing into truly functional cholinergic neurons. And we found that in the Down syndrome samples, there were more of the mature neurons, meaning, or we interpret this as the neurons are maturing faster in Down syndrome. And this is also shown by the GO terms that we found that were associated with these different subtypes. And this fits with what has been shown and postulated, that there is a precocious maturation of neurons in Down syndrome. We also looked at we looked at what the differential genes were associated with in the cholinergic neurons, and we found that most of the upregulated genes in the mature basal forebrain cholinergic neurons were associated with OXPHOS, suggesting that there is an increase in energy, or there's a change in energy metabolism, specifically in the upregulation of glycolysis and oxidative phosphorylation genes. And this may be indicating that these neurons are starting to shift towards glycolysis as the primary source for energy production. And then that's a shift that's often linked to mitochondrial dysfunction and several neurodegenerative diseases. And then when we do gene concept analysis, we find that these pathways, are also associated with other neurodegenerative disorders like Alzheimer's disease and Parkinson's disease. And so we hypothesize that this sustained oxidative stress predisposes these cholinergic neurons to early degeneration in Down syndrome, and we're trying to test that with our human stem cell model. In addition to the neurons, I also want to highlight what we are seeing in other cell types, primarily the astrocytes and microglia of the brain. And our results indicate that a large proportion of astrocytes in the Down syndrome basal forebrain express molecular markers that are associated with reactivity. That means these, and we confirmed the presence of reactive astrocytes in the basal forebrain using immunofluorescence. And you can see here that these transcriptomically, the astrocytes in Down syndrome express reactive markers, shown here with HSPB1 and CRY-AB. And you can see this with immunofluorescence for GFAP, another marker of reactivity in brain sections. We saw a similar activation in microglia in the Down syndrome brain, and this microglia are the immune cells of the brain. They are resident immune cells, so these are, based on our analysis, these are not infiltrating microglia, but they are resident brain microglia, and our results indicate a large proportion of microglia in Down syndrome are activated, again, as indicated by the increased expression of complement and HLA genes shown here. And we also validated this in the tissue using immunofluorescence for IBA1, which is a marker of activated microglia. So we think that activation of microglia and astrocytes and the consequent production of pro-inflammatory cytokines might trigger neuroinflammation. And these cells, both the astrocytes and the microglia, normally function to maintain neuronal homeostasis. However, chronic neuroinflammation, as we're seeing here, can lead to neuronal damage. And so this is something we were really surprised to find, again, at this early age. And so to summarize, what we found in the basal forebrain is that we see a precocious development or maturation of the cholinergic neurons, again, that aligns with a general hypothesis that we have about developmental heterochrony in the brain and other organs. And we see this dysregulated energy metabolism, increased oxidative stress. And again, we think this is an early stage of shifting towards glycolysis. And finally, we see reactive astrocytes and activated microglia, suggesting that there is neuroinflammation in the brain at birth. Now I want to switch to the prefrontal cortex. This is arguably a more interesting area of the brain because this is the part of the brain that really makes us human and enables us to do human-specific functions like executive functioning, working memory, and it is one of the most complex and heavily interconnected regions of the brain, and it is also often impacted or thought to be impacted in many neuropsychiatric disorders. So we did a similar study from using the dorsolateral prefrontal cortex from postmortem tissue from 0 to 3-year-old samples, and we have 5 in each, and we tried to match for age, sex, and PMI, and this is the number of cells. We did single nuclei RNA and ATAC-seq, and again, this was with Kalpana and also with Ryan Risgaard, an MD PhD student who also just recently graduated. The first thing that we saw when we looked at the cell composition was we saw, surprisingly, an increase in excitatory neurons and a decrease in astrocytes. So again, this mimics what we see in the basal forebrain, more neurons, less astrocytes. And again, contrary to many historical data, including our own, that there's actually more astrocytes and fewer neurons. So we feel like using the transcriptome, we can maybe better identify these different cell types and have a more accurate definition of the cell composition. When we looked primarily at the excitatory neurons, so these are the layer 2-3 excitatory neurons that are the main functional output of the cortex, we can see that the go terms associated with the upregulated genes in Down syndrome are primarily in metabolism and biological processes related to metabolism and immune response. And you can see this by looking at specific genes associated with metabolism, reactive oxygen, even apoptosis are increased in our Down syndrome samples and actually increased quite a bit. This aligns with the reactive astrocytes and the reactive microglia that we also see in the prefrontal cortex, similar to what we saw in the basal forebrain, but actually in the prefrontal cortex, the microglia are really all activated, suggesting really a pretty profound neuroinflammatory signature in the prefrontal cortex at birth. And so just to summarize our results from the prefrontal cortex, I didn't talk to you about developmental heterochrony, but we also see our data along with some prenatal data suggests that that's also happening. But primarily we see a dysregulated energy metabolism and this neuroinflammation at birth. And so we are, I will say, we were surprised to see such a robust inflammatory response at such an early age, and really makes us reconsider how we think about neurodevelopment versus neurodegeneration in Down syndrome. And so we think of this as more of a continuum that is probably starting early in prenatal development and is just continuing throughout the lifespan. And neuroinflammation in particular, as has been postulated by Joaquin's group, and is probably has a huge impact on what is happening in Down syndrome as far as the clinical symptoms, but we were surprised to see it so early and in the brain. And so going forward, these data shift the temporal onset of Down syndrome-associated neurodegenerative processes to early postnatal. We also identify a potential therapeutic window in early childhood where we can consider some interventions that may alleviate some of the neuroinflammation and hopefully lead to some better outcomes. We also have some similar questions to what Micah suggested. What comes first, the chicken or the egg? Does neuron dysfunction precede neuroinflammation, or is it the glial cells, the astrocytes and the microglia that are driving this? And we are using our human stem cell modeling to try and test this to try and test the contributions of the different cells, using, which I think is really an advantage of human stem cell modeling, is that you can mix and match different cells. You can mix and match trisomy and control cells and see how you get the inflammation. And we do see neuroinflammation in vitro. We also are really interested in how oxidative stress and neuroinflammation, which are two different pathological events, how they may be linked, and again, which one might happen first. And finally, I want to say that this data has all been deposited with the fantastic help from the Data Coordinating Center. They helped us deposit the data in dbGaP, and the data can also be accessed through the data hub that Joaquin talked about. And we also have some ways, some apps to interactively visualize the data if you don't want to go to the raw data. So I'll put those in chat afterwards. And so finally, I just want to thank the funding and the people who did this. I mentioned most of the people, and again, my fantastic collaborators. And I'd be happy to take any questions. Thank you.
Thank you, Anita. That was great. Yeah, questions, anybody? Feel free to enter in the chat or unmute yourself. There's a question, Anita, from Bruna Sampieri from Brazil. Hi, Bruna. About the causes of death for the causes of death for the individuals with Down syndrome.
Oh, yes. Yes. Yeah, that's a great question. What were the causes of death? They were varied, they were varied, and we had a pretty small sample size, right? So they were varied. Some we don't actually have a cause of death. Some were like drowning or something like that. But we used cause of death as a covariate to see if that made a difference, and it didn't, but it might be that we're kind of underpowered. All the covariates we tried did not show some positive association, like postmortem interval, things like that, for any of our studies. We usually do that first. But again, we have a pretty low sample size, which is an issue. I mean, I think that, it's good that there aren't that many early postmortem brain samples from individuals and controls with Down syndrome. So, but that we are really limited by that.
Thank you, Anita.
Thank you, Anita, for a great presentation. Yeah, it's very interesting study as well. So I just want to mention that I just want to check with you. Have you checked the GTEx, the Common Fund GTEx data set to see if there are controls or participants with Down syndrome the data you can use?
Yeah, I'm going to just stop this a minute. That's a good question. We are starting to do that now. So this, as I said, a lot of this data is really fragmented, right? We have some, somebody else has some, and I think that is really the power of the Data Coordinating Center. We're right now trying to synthesize some of the other data that's published, including going to GTEx as well. So we haven't done that yet. Yeah, that's good.
Maybe you should chime in on Wei-Ching's comment. The other resource that's really exciting, and we're looking for ways from the Data Hub perspective to interact with it, is the developmental GTEx, or the DGTEx that just came out, as a framework. And then another program that I think we're currently trying to assess relatedness is the SMAT program. This is another Office Director led program that looks at somatic variation across a sort of normal biological context in the CNS, and wondering if there are differences, across conditions in that setting.
Yeah, that's great. We have not used those, but what we have done is we use the PsycEncode, right? So that has the developmental. So
yeah, the DGTEx, I think, just released some of the very first data sets, and they have a CNS cohort in that setting.
Yeah. That's good. Thank you. That's good information. And thank you, Pierrette, for putting the link in the chat to the data. And thanks for all your help.
I should point out, Anita, that Nicole West, if I remember correctly, was and a student in the DS3 course, the summer course for data science under the auspices of the Data Coordinating Center.
Yeah, that's great. I should have pointed that out. Both Nicole and Kalpana were, and that really helped them deal with this data. Kalpana, of course, is a biostatistician, so he had a little more background, but Nicole went in cold, and going to DS3 really helped her. So thank you for pointing that out.
You may have the distinct honor of having the most student in DS3 over the years. Yeah.
Well, that's good. I'm happy.
Yeah, your
and I
lab
have
always has
more.
very strong candidates.
Thanks.
Well, very good. If there are no further questions or comments, we'll wrap it up here. Thank you, the speakers today, Dr. Bhattacharya and Donan, for two great presentations. And we'll see you at the next Data Hub webinar in the fall. Thank you, everybody. Have a great week.
Great. Thank you.
Thanks, everybody.